This is part 2 of the RNAseq Lab. I will talk about the differential expression analysis at gene level. So first, let's go to a classroom at OSC. class.osc.edu. You can log in with your OSC account. And this is different from on-demand. This is just for classroom usage. And I already installed all the required R package in the RStudio environment. You can launch it by Interactive Apps, RStudio Server. And the class material is in BMI 5730. And the number of hours, we can require 1 or 2 hours. And the launch. So similarly, you can manage your files in this home directory or our classroom directory. So once your first time log in from this class.osc.edu website, there will be one folder created automatically in your home directory under Users. So this folder name is OSC Classes. And then it will automatically create a BMI 5730 folder within that folder. And you will have the materials and the R. So that's the material I put in my folder. And you need to copy paste those materials from our classroom folder under FS project. And this is also has a folder BMI and materials. You need to copy all these materials from this BMI 5730 folder under project to your own home folder in this OSC classes. So I will show you a way to copy paste this material. Okay, so now my job is ready. I can connect to the RStudio server. Okay, once you first connect to the RStudio server, you will be at your home directory, which is this absolute path. And you can go to the terminal. And then you can copy materials. You can first list all the material in this folder, and then you can copy everything. In this folder to your current home directory. Yeah, I will not copy them since I already have it, but you can copy all of them this way. Okay. So let me open a file in my home directory. I have an install package .r file. This listed how I installed all the required package we're gonna use in this lab in our classroom RStudio environment. Sometimes you will encounter a lot of problem when you try to install DESeq2 in your own laptop or your own computer because some required packages are not successfully installed. So you have to look at the error message and specify each of them and install them one by one. Okay. So today we will use the R Studio to do some gene level exploratory analysis and differential expression analysis for the RNA-seq data. So you can, after you copy those files into your own directory, you can open it and go through it line by line. So let me first start with load this library, Lima HR and the DPLYR. This library, the last library, is the one I like to use to manipulate data. So this count table is the output result from the full data sets from our RNAseq Lab Part 1. So it's located here. If you look at the full data set, it's in the full data sets post run. This RNAseq reports folder. It has all the summary reports from the RNAseq and the feature count. And the important one we're going to use for downstream analysis is this one, RNAseq feature counts. That has all the recounts for each sample, total 14 samples. Another one we're going to use is the RNAseq feature counts length. This one has gene length for each gene, and this we can use it for calculation of FPKM. And some other data we need is in the reference file. That is our annotation file, this one. It's not the whole annotation file, it's just the information we need. This gene ID and the gene name. This file is grabbed from this GTF file. Take a look. Yeah, it's the first column is the gene ID, the ensemble gene ID. The second column is the symbol gene name. Okay. So I already copied all the required data set into this materials folder. Oh, one more thing we need is the sample information. This one tells us out of the 14 samples, 7 of them belong to the pre-group, and 7 of them belong to the post-group. And the sample ID, they name them equally for the paired sample. R1 is same as R1. So this way it will give the design matrix the paired sample information. Okay, so first let's read the table in. And here we set the string as factor as false, and the header is true because this RNAseq feature count, we can look at the count table. It do have the header line. That's the sample name. And there is an X here, we don't like it, so let's rename the column name. First column name, instead of X, let's name it as gene ID. Okay. So the requirement for the input for the Lima package is we have to make the count table instead of a data frame, but it has to be a matrix. So DM is the matrix. So we remove the first column and just keep the recount from the rest of the 14 samples. And we give the matrix a row name. The row name is equal to the gene ID. And next, let's read in the sample information. And the row name of the sample information is the same as the first column of the sample info. The first column of the sample info is the sample name. So this way, the row name instead of 1, 2, 3 is equal to the sample name now. And now the sample names, let's make a factor with 7 levels. And the group is also a factor with 2 levels, post and pre. Now let's read in the gene annotation file. Gene anno with 2 variables. Let's give it the column name. The first column, gene ID. Second column, gene symbol. And then let's only keep the ID, keep the gene ID from the count table, because this gene anno file has it also has 58,000 observations, 58,000 genes. It's the same as the count table, okay? But the order is different. The gene name, gene anno order is different from the count table order. So let's grab the ID from the the count table, and then merge it by gene ID. So this way, the gene ID is all ranked by as the same order as the count table. And then let's read in the gene length and the left join the gene data frame and the gene length data frame, and specify the merged ID is gene ID equal to gene dash ID. Genes. So now the length is merged to this table. And this is our annotation file for gene. And now we can create this DGE list for Lima and specify that the count is equal to this data matrix, and the group information is in the group factor, and the gene annotation file is in the genes data frame, and the samples information is in the sample names factor. So now let's look at what this X samples look like. It has 14 rows. It only shows the top 10 rows, and has 2 group pre and post, and the library size for each sample. And the normalization factor, we haven't normalized the data yet, so it's all by default 1. And the sample name R1 to R7. And then next, let's do some transformation. So the first step is to do transformation from the raw scale. For differential expression related analysis, gene expression is rarely considered at the level of raw counts. And we will do the normalization for library size and also others. I talked about this part in the RNAseq lecture. There are totally 3 different types of normalization. Normalized for gene length, for library size, and also for RNA composition. And the popular transformation include count per million, log 2 count per million. That's the one we're gonna use later on. And that's the most common one used for differential expression gene analysis. FPKM is good for just compare genes, different genes within one sample. But when we compare the same gene across different conditions, since the same gene will have the same gene length, so we don't need to normalize for gene length. So here, let's transform it to count per million and further transform it to log count per million. And the second step is to filter lowly expressed genes. There are different ways for filtering. So here I will show two ways. Let's first check the total counts. So how many genes that has the recounts that is equal to are 0 across all the condition, all the 14 samples. There are 17,000 genes. So the absent gene across all the 14 samples are 29%. So we use this filter by expression function in the HR package to automatically filter genes. And you can look at, there are very detailed description of this function in the HR package. You can look at it carefully. And if we run this, we can see Y is only the the left gene. We only keep the 19,000 genes out of initially 58,000 gene. So now let's look at Y. This information is still the same. The samples information is still the same. Just the number of genes are reduced from 58,000 to only 19,000. So we can plot the distribution of the gene and to compare what it look like before and after filtering. This is the raw data before filtering. We have a huge peak here for lowly expressed genes. The x-axis is log CPM. And after filtering, the huge peak disappeared, and this distribution is, the data is more normal distributed. And we can have a loose filtering threshold. People sometimes also use their own threshold, but this is quite arbitrary. For example, people can calculate, this means apply the function to count how many cells for this row, for each row of this DM matrix. That means for each gene. Let's count how many samples have the recounts greater or equal to 3. And if we table this, we can see there are 22,000 genes that has no sample greater or equal than 3, and we can select. We want this to be no sample, the cutting threshold, or 1 sample, 2 sample, 3 sample, and here we just arbitrarily choose. Okay, I want it has at least more than 3 samples that have those minimum recount greater or equal than 3. And the DM2, we can look at it. It has 30,000, about 30,000 genes left. So this filtering criteria is relatively loose compared to the first one. So I will not continue with this, because when I look at the plot, when we check the plot, you can see the data is not clean yet. You still have a large peak at the lowly expressed gene. So this is just to show you an option. If you want to do, if you want to keep more gene in your gene list, you can do it this way. The third step is normalization. It's very important to do normalization, and there is a popular way and a very good way to do it by this calculate normalization factor function, and you can choose different methods, and the TMM is a very popular method and a very good one. So now let's look at the normalization factor. Yeah, it's here instead of 1. Now some samples has 0.9, some sample has 1.08 normalization factor. And let's visually look at what it look like before and after normalization. For unnormalized data, this data set is okay. You don't see some outlier that its distribution is too off. But for some data, you can see 1 or 2 sample that is way too off. After normalization, you can see the median is more like at the same level for each sample. And here I also provided the code to calculate RPKM. We didn't use it here in this in the following analysis, but this is just provide you a way. Some biologist people, they want to know the RPKM, so we can calculate it this way. The fourth step is to do some clustering of the samples. So one way is called MDS plot, multi-dimensional scaling plot. So this plot shows the similarity and dissimilarity between samples in an unsupervised manner. So ideally, samples should cluster well within the primary condition of interest. Here is pre and post drug treatment. And if any sample staying far from its group, this could be identified as an outlier. So let's look at the plot. I plotted it in 2 ways. The first is by the sample groups. So here is the leading log fold change at dimension 1, and the y-axis is the leading log fold change at dimension 2. We hope to see that the pre and post group separated pretty well at the major loading log fold change dimension 1, but it's not separated perfectly. This post sample mixed up with the pre sample, and also this post sample also kind of mixed up with the pre sample. But overall speaking, the separation is okay. And if we want to know which sample is mixed up, we can look at the sample IDs. We can also do the heat map to show the Euclidean distance between samples. Here I use a function in DESeq 2 to show the sample distance. Okay, so this is just to calculate the sample distance by the function in RNAseq 2. And next, I will use the p-heatmap library to do the heat map plot. The first one is based on the group. I cannot know whether it's pre or post, so I plot the second heat map. So similar as what the MDS plot show, there are some pre-samples mixed up, pre and post samples mixed up. And if we look at whether they belong to the same patients, and yes, they belong to the same patients. So here, the biological differences is more significant compared to the pre and post drug treatment differences. And another way to visualize sample to sample distance is principal component analysis, PCA. In this analysis, the data points are projected onto a 2D plane, so they spread out in the two directions that can explain the most important variance. So here, the x-axis is principal component 1. It can explain 32% of the total variance. Actually, this is not a very good PCA plot. Sometimes a good one we expecting to see over 50% of the variance can be explained by PC1. And the PC2 can explain 18% of the total variance. And similarly as what the heat map and the MDS plot show, some data points of the pre and the post mixed up, and the separation is just okay, not perfect. So step 5 is differential expression analysis. In this study, our main interest is to see which genes are expressed at different levels between the post-treatment group versus pre-treatment group. And there are usually many ways to set up a design matrix. Here I will show some different way. The first one, if we do not consider the paired sample comparison, which is not very common. I would say most of the experiments design are not designed for paired sample. So if just we want to compare two groups, then we can do the model matrix, this tilde group. In this way, the design matrix will look like this. The intercept represent the mean of the pre-group, and the group post here actually represent the difference between the post group versus pre-group. So it only exists in the last 7 samples. And this approach is also called the treatment contrast parameterization. For the second approach, we can add a 0 plus before the group. In this way, it removes the intercept from the group, and then the contrast, the design matrix will look like this way. So for the first group, for the first 7 samples, there is the 1 disappeared here because the first column is the mean represent the first coefficient represent the mean expression level of the post group, and the second column represents the mean expression level of the pre-group. And this way, we need to make a contrast matrix, post versus pre, equal to the group post minus group pre, in order to make the pairwise comparison. So I usually use this way because I feel it's more convenient to make contrast if we do not consider the intercept. And since we have the pre and post samples from the same patient here, it's better for us to do the paired sample comparison. So here we need to add the sample information in this design matrix. And also similarly, without the 0, it is a matrix with the group intercept, and with 0, it's a design matrix without the group intercept. And let's do this, the design 4. Let me see, probably I need to run this first. Let it know what is a sample stand for. And then this one. Okay. So this is the final design matrix we need to use. The first one is this one, yeah. The first one is the mean value of the group pre, and then is the mean value of group post. That's what we are interested. But while they compare this, they will also consider the paired sample information. That two samples are from the R2 and the 2 samples are from R3 extra. So then we can create a contrast for what we are most interested in, the post versus pre. And then we need to do the lemma-Wu transformation to apply it on the RNA-seq count data. The lemma is developed for microarray data analysis. So when we want to use the lemma linear model on the RNA-seq count data, we have to do the Wu transformation. This is very simple, just the Wu and the data Y and the design matrix. And let's take a look. This way it will just plot I say plot one by one. So I want it to show side by side, so have to run this code chunk together. So the first one shows the mean variance trend of the RNA-seq data recount before the Wu transformation. We can see that the variance is not stable. It's decreased as the log2 count increasing. So for the recount with the small number expression level, for the genes with low expression level, the variance is very big. And after the Wu transformation, we stabilize this portion, the variance for lowly expressed gene, and the variance now is independent, is quite consistent. no matter the gene expression level. And after Wu transformation, we need to do the linear modeling. This is accomplished by the lm-fit command and the contrast fit command. And then after that, we need to do empirical Bayesian moderation. This is accomplished by the e-Bayesian command. In this step, the empirical Bayesian model will borrow information across all the gene to give it a better estimation of the gene level variance. So this one, I just, because most of the time, I usually do not just make a one contrast. I will do multiple pairwise comparison between different groups. But in this case, we are just working on an easy case, just one contrast. So this for loop actually is quite redundant here. But I still keep it here. And you can use the top table of eFit to show all the results of the data. Let's look at this TMP. That's your final results. Has gene ID, gene symbol, and the length of the gene, and the log fold change value, and the average expression value, and the t-statistic, p-value, adjusted p-value. And I also calculated this fold change as the last column. So you can just save this data for this 19,800 genes. And for a quick summary, we can run this command summary decide test. So this will tell us out of this 19,800 genes, 15,000 of them are not significantly differential expressed, and 17,064 genes are upregulated, and 2,783 genes are downregulated. Compare post versus pre. And for a stricter definition of insignificant, we can not only use p-value, adjusted p-value as a cutting threshold, we can also require the fold change greater than 1.5. Then that is the log fold change equal to log 2 based and 1.5 fold change. So this way, we can see the number of significantly differential expressed genes are significantly reduced from thousands of genes to only 50 or 300. And then we can do some MD plot and volcano plot to visualize those differential expressed genes. And here, I think I used some this function from Lima and RNAseq 2. Let's run this code. So this is the MD plot. The blue dots are the downregulated gene in post sample compared to pre, and the red dots are the upregulated genes in post sample. Here is the volcano plot. Those are the downregulated genes, and I think it didn't meet the threshold for this plot function, volcano plot function. So there is no upregulated genes highlighted in this plot. You can tune all those parameters. And we can also visualize the top 50 genes as ranked by the adjusted p-value from the comparison. And a heat map is created based on these genes. Again, I used the p-heat map package. So here are the top 50 upregulated genes and the top 50 downregulated genes. And I also ask the heat map to cluster based on the similarity between the pre and the post sample. So there are some clustering. So that's all for this this lab. And I also list some section information, and here is the reference material I used online. Also, I think I generated a HTML file. You can also look at this file while you are doing this lab.